Thanks very much. I'm not sure this is punishment to me or punishment to you guys, but I'm really actually looking forward to sharing some of the thought processes that have been going on in our lab and some of our recent data. So for those of us that were mentioned earlier in terms of interventional cardiology, yes, we do have some passion in vascular biology, and we have at least 3 of us here. So I spend some of my clinical time in the cath lab, and what on earth do I care about in terms of oxidative signalling? We have known for a long time that redox signalling actually plays an integrating role in almost all of the known risk factors that we've got driving cardiovascular disease, but also probably as a common mediator of a lot of the unknown ones that we've touched on. This is actually from this morning's Daily Telegraph. I don't know if many of you have met Scott, who's actually Bob Graham's major right-hand man in the lab at Victor Chang. But he is a chap who has one of these traditional kind of zero risk factor profiles, rides 40 kilometres a day on his bike, had a bit of indigestion, ended up at Hornsby Hospital, and came through to North Shore, where he promptly arrested on our cath lab table. And we basically, when he woke up, he's sitting there going, why me, why me? And I think this is basically a story that we've already touched on in terms of the increasing group of people that don't have any of these traditional risk factors. And perhaps by improving our understanding of dysregulated redox signalling, we might actually get some insight to it. That, along with obviously the other interesting opportunities that we've discussed today. So this is just data from the cath lab at North Shore, where we see over the last decade or so, 15% having 0 of the traditional risk factors. We obviously don't measure LPA or HDL as raw numbers in this, and this is just from a practical experience. And the total percentage of them that have 0 or 1 risk factors is obviously pretty high over that decade. But there's actually this quite strong increase over the last decade as well. And this might reflect, obviously, improved primary prevention. It might improve something to do with the changing epidemiology around our hospital. But it certainly raises interesting opportunities for novel discoveries. We certainly don't know everything about cardiovascular disease. So back to redox signalling, clinical trials have obviously also hampered the kind of hypothesis about redox having a role, but I don't think that should completely scupper it. Redox signalling is very complicated. We're not just a big bag of redox kind of species that just we can measure, willy-nilly. Compartmentalisation is one aspect, and this is obviously 2 of the important compartments that we'll talk about today, one being the caveoli and another being the mitochondria. The other thing that's actually pretty important to consider when you talk about clinical trials is we're pretty bad at measuring dysregulated redox. So if you're actually going to be, if you imagine trying to set up a blood pressure study with a new antihypertensive, you'd like to be able to actually measure it, at least at the beginning and in response to the treatments. I'm not going to be talking about the oxidisation of LDL. I'm actually going to be focusing on intracellular redox signalling and the dysregulation of that occurs in a number of disease states. The mitochondria, I'll give one slide of. I am not an expert in redox signalling related to the mitochondria, but during the generation of ATP, the flow of electrons obviously can be involved in a positive way, but it also can culminate in the reduction of culminating in the reduction of oxygen to water. But it also, in the process of this, depending on the voltage of the mitochondria and a number of other factors, can release superoxide. How that then regulates what's happening in the cytoplasm and perhaps in the membrane is less well understood, particularly as superoxide actually doesn't particularly travel very well, and it requires sort of catalase to turn it into hydrogen peroxide and then transport out into the cytoplasm. Now I'm getting to the compartment that I want to share with you how amazing this little guy is. So the caveoli we've heard about today as the home of endothelial nitric oxide synthase. It also is the home of NADPH oxidase. And these 2 enzymes are some of the 2 biggest producers of oxidative species. NADPH oxidase, that's its job, and that's often receptor coupled in the caveoli. But eNOS itself, as I mentioned, is not only a producer of the good guy, it's also a producer of a large amount of the superoxide. And importantly, it's doing it in a vicinity of a whole bunch of redox-sensitive proteins. And so the caveoli acts as a really nice signalling microdomain. It couples receptors to these enzymes that then produce these redox species in the vicinity of a huge number of proteins, of which about 80% are thought to be redox sensitive. So glutathiolation is akin to phosphorylation. It's a mixed disulfide bond that forms under conditions of elevated ROS. Similar to phosphorylation, it actually has an adduct that's a negative charge, and it's a similar mass and has steric effects on the protein's function. Importantly, it's actually enzymatically reversible. So who are the players that are controlling the switch of redox signalling and dysregulation in the caveoli? NADPH oxidase we've touched on. This, when it's activated, for instance, by angiotensin binding at the AT1 receptor, leads to gathering of all of the subunits in the caveoli. An important fact here is that the superoxide is then produced into the extracellular domain of the caveoli. We all think of the caveoli as the signalling domain where things are happening here, but in fact it's all the proteins that are scaffolded onto the outside. Superoxide then is involved in somehow getting back in and modifying these associated proteins. That's probably via aquaporin, which I suspect plays a very important role here and has the capacity to carry hydrogen peroxide across the membrane. So eNOS obviously does lots of great things, but this key paper that I touched on in a question earlier found that one of the key molecular mechanisms of uncoupling of eNOS is glutathiolation of these two cysteines. That's in the reductase domain. It results in a reduction in NO production by 70% and an increase in superoxide by 5-fold. So if we can find ways of modifying this, this is actually really an important factor. Now, how much of the superoxide produced in the caveoli microdomain in response to something like angiotensin, which is a major driver of dysregulated redox in the cell, how much of it comes from NADPH oxidase and how much of it actually comes from eNOS being uncoupled? We all know that if you block NADPH oxidase, you block most of the production of superoxide. But that doesn't mean that the downstream eNOS uncoupling could actually be a key mediator, which would put a whole new light on these particular cysteines. And because of this discovery by Chen et al., we could actually now mutate the cysteines and see exactly the answer to this question. And so this is some work we did with colleagues at the HRI, where we found that in cells, Ang2 indeed caused an increase in the level of glutathiolation of eNOS, which is the molecular mechanism of its uncoupling. That was not surprisingly associated with a decrease in nitric oxide. And this is causing an increase in superoxide as measured by HPLC techniques. But what is fascinating is in these cells that are expressing the double cis mutant that is not able to be glutathionated, we almost completely abolish the increase in superoxide seen in response to Ang2. So this really highlights this is a really important new mediator of dysregulated redox signalling in the environment of over 200 proteins, of which 80% are redox sensitive. So just to say here that this little puff of superoxide that's produced by NADPH oxidase definitely has a role, because we know if we block this, it blocks this. But in fact, we suspect these guys are playing a huge role. So this makes the caveoli suddenly a big furnace where you've got a bunch of key proteins controlling intracellular electrolytes and all sorts of signalling suddenly exposed to this explosion. Now, we did mention the sodium-potassium pump, and it is actually a fascinating molecule. It's a pretty important one, using between 25 and 50% of cell ATP, and that's setting up all sorts of key gradients for sodium and calcium handling, as well as a number of other aspects. This is important in terms of its regulation affecting systolic and diastolic function, but also as a driver of long-term signalling changes. This is some work that I've done back in a few years ago now, but we actually identified a single cysteine on the beta subunit that was actually mediating this. And when it was glutathionated, it basically inhibited the total turnaround of the pump, and it was unable to actually turn over between its E1 and its E2 conformational state. This is the the dynamic work that demonstrated that, where we actually found that this particular transformation or conformational change was the one that was blocked by this steric addition of the glutathione, which, don't forget, is enzymatically reversible, occurring here. So this is a really interesting way to start thinking about this important microdomain. So the sodium-potassium pump is just one example of a whole bunch of really important molecules that are sitting right next to this little explosion of enos. We've done since a whole bunch of work. What can we do to try to reduce this? Interestingly, one of the best antioxidants is actually all the things that we know work in cardiovascular disease. So if you block the angiotensin receptor, you dramatically decrease enos glutathiolation and also the sodium pump's redox inhibition. Similarly, if you block the beta-1 receptor, you stimulate the NPRC receptor, or indeed the beta-3 receptor, which is here, which is a beta-3 adrenergic receptor. These all lead to various intracellular signalling changes that result in a decrease in enos glutathiolation and also the sodium pump. However, if you give an animal vitamin E or you give a human vitamin E, we don't expect and haven't demonstrated any ability to change any of this. And so that's a really important point. Other than the proven therapies, are there other ways that we can think of to try and target this important molecular event? This is just demonstrating the beta-3 adrenergic receptor, which we know increases enos activity and is in fact used as a stimulant for enos. In a novel model of diabetes that my PhD student Kayvan Karimi developed, where we used a peptide inhibitor to the insulin receptor, it's really a hypoglycemic model rather than a diabetic model, because you're obviously blocking the insulin signalling within the cell. But we saw here that the enos uncoupling or enos glutathiolation was dramatically increased in hypoglycemia. But by infusing the animal with a beta-3 agonist, we decreased that enos glutathiolation and uncoupling significantly. We had a corresponding dramatic decrease in the superoxide production in the aorta of these animals, an increase in the nitric oxide production, and the corresponding increase in the improved vascular reactivity. So of those 250 identified proteins that live in the caveoli, of which 80% are thought to be redox sensitive, about 1% to 2% are thought to be involved in protecting those proteins against redox modification. So glutaredoxin 1 is classically thought to be a cytoplasmic enzyme and is the major specific deglutathiolinator in the cell. But what regulates or somehow gets glutaredoxin to the caveoli? We've certainly seen that it gets there in partners with proteins that are living there. And in fact, in parallel with the decrease in the glutathiolation, in this case of the beta-1 subunit, we saw a improvement in the co-immunoprecipitation of glutaredoxin with both beta-1 and enos. So it's suggesting that something during this treatment is actually increasing the ability of glutaredoxin to actually get to its target protein and protect it. And we're still fascinated by what's actually doing that, but obviously that's an important therapeutic pathway to work on. This was picked up in an AJP editorial by Stephen Black, who was really also fascinated by this data, which was actually buried in panel F of figure 8 or something. But really the fact that GRX1 is predominantly a cytosolic enzyme, what's regulating it to actually have this key role in the caveoli to actually protect these proteins and reduce their redox inhibition. I don't know if any of you followed the bardoxolone literature, but in terms of efforts to try to improve cellular redox state, NRF2 dependent, our drugs acting on in the NRF2 pathway, which promote a whole bunch of redox protective mechanisms, have been trialled and actually were successful in a phase 2 trial in impaired renal function patients. For some reason, they hadn't actually followed the cardiovascular pathway, so I was actually working with Reata, who was the company that was developing it, and we were trialling it in various cardiovascular models. And indeed, in this particular case, what we saw, this is an ischemia reperfusion model, and a long-term, it's actually post-infarct, following out over time both cardiac function as well as protein changes. And what we see here is that the glutathiolation of enos is actually increased in the peri-infarct zone and indeed the remote zone, which I won't show here. But that was, at 28 days, quite different in the group that received the bardoxolone equivalent. We similarly see an increase in the association of glutaredoxin with enos. And from a functional point of view, obviously not a causal relationship, but a fascinating suggestion. What we see here, I'll just focus mostly on the end-systolic area, which is one of the most important prognostic variants post-infarct. Not surprisingly, we see in the main, in the control group here, the vehicle, an increase in the end-systolic area. And this was quite dramatically improved in those that had received the bardoxolone equivalent and in association with these molecular improvements. So this is something that actually, given it's already safe in humans to a relative degree, I think is actually a potential treatment that we should explore in cardiovascular side of things in humans. Now, just in terms of competing with complex diagrams, these are the proteins that are in the caveoli and their relationship, highlighting here again just a small percentage of them that are actually involved in redox protection. We were fascinated by a particular protein called FXYD, and I'll just skip to the point in the sense that this protein's only 14 kilodalton and is thought to play a role in regulating phosphorylation of the sodium pump only. We've previously shown that it did protect the sodium pump from glutathiolation, and it's got a couple of candidate cysteines that we proved were doing that. But fascinatingly, given enos, we think, is the switch, we found that FIXAD1 actually co-immunoprecipitates with enos and dramatically protects it. So when you silence it, it causes an increase in the glutathiolation, which is associated with a dramatic decrease in nitric oxide production. So this is suggesting that FIXAD1 has a new role in actually protecting enos, and this has obvious therapeutic potential. In vivo, in terms of vascular function, we found in the knockout mouse a dramatic decrease in endothelial-dependent relaxation as measured in some in vivo vascular experiments. This is just highlighting a potential way in which we see FXYD improving vascular function by somehow protecting against the glutathiolation of these cysteines. So, given we're talking about biomarkers, the key point is that no redox biomarker does much other than associate very poorly with disease, but doesn't actually, most of its R values are about 0.2 in terms of associated with changes of redox signalling in the vessel wall. We're trying to find better ones. We've had some, by measuring the levels of glutathiolation of the sodium pump's beta-1 subunit in the erythrocyte, we've actually found that the compartmentalised signalling is actually preserved and actually correlates beautifully with what's actually happening in the vessel wall. So if you're looking at measures of dysregulated redox signalling in the endothelium of a human, where we see a strong correlation in vessels that we explant from humans of this measure with redox dysregulation in the vessel wall. The R value for that is over 0.85 compared to some of these other markers which are about under 0.2. Obviously, I'll skip over this. To summarise, and to think about that drink that was mentioned earlier, the caveoli are a fascinating microdomain that are home to over 250 proteins, many of which are ROS regulated. The fact that they're sitting right next to two of the biggest producers of ROS is really a tantalising opportunity in terms of how do we regulate these and how do we protect the proteins around it better. Treatments that are well proven in cardiovascular disease are associated with decreased caveolar redox modification. We need to find some more specific attempts before we actually try to target the next redox protective approach. And I think we should probably leave it at that, except to thank the large amount of collaborators and funding.
Thanks very much.